Yeah, and this is called a condensation reaction because water comes out. This is water right here. Right, so that's to make it bigger. What about to break it down? If you want to break it down, what do you got to do for these molecules? To break down, to make smaller. So this is if you have these kinds of molecules strung together, to make them smaller, to break down. To break down, go ahead and give it to me. Break down. Okay, molecule and molecule are together. It's the exact reverse of this. So you've got this molecule. It could be one carbohydrate connected to another carbohydrate. Okay, now water comes in. Water comes in and says, what's up with this? You guys break apart. I don't like you hanging out together. Right? So this water comes in and says, you two get away from each other, and leaves one of the molecules with an H and the other one with an OH. I'll draw the molecules again over here. So this is the first step over here, the water's coming in, and then afterwards this is the post. This is the after picture. Here's a molecule there, there's a molecule there, there's a molecule there. They were bonded together. Now they're broken apart. Oh, so sad. And the molecules are now split apart, and they're split apart by water. So we have a word that means water splitting, which is hydrolysis. Hydrolysis, in other words. Some people say hydrolysis. I don't. I say hydrolysis. This is to break down. This is a hydrolysis reaction. This whole process right here, water coming through, splitting it apart. So know that as well. So I've gone into some detail there about the different macromolecules and then the reactions to make them bigger, to make them smaller. Right. Let's go on to water really quickly. So I'm going to give a brand new page right here. But remember this up here, those steps. Okay, new. Water and osmosis, okay, these kinds of transports and so forth. Know what diffusion means? So know these terms. Know the term diffusion. Know the term osmosis. Know the term active transport. Know the term passive transport. Okay. A good activity to kind of distinguish all of these is to say, try to make like 3 or 4 sentences in which you describe how these things are different from each other. So just use these words more comfortably. Say, I don't know, osmosis is an example of diffusion, but it uses just water. Passive transport uses the diffusion gradient. Oh, gradient. Ooh, you know what that means, gradient. Okay, gradient, I'll just tell you right now what that means. That means that there is we call it concentration gradient, actually, concentration gradient. Concentration gradient is just where there's one area that's really dense and packed together, and then it starts to spread out more. Okay, so if this is like a container here, we'd say this container has a concentration gradient. There is an area where, I'm just drawing lots of dots here. There's an area where these dots are very packed together, that's over here. And there's also an area where they're spread out. So there is a gradient along this container here, a concentration gradient. You know, there's a higher concentration here and a lower concentration here, and it's kind of smoothly distributed. All right. Okay, so read those again, make sure you know what these ideas are and how to distinguish them, what requires energy and so forth, what requires proteins, because sometimes these require proteins to happen. When you're pumping something from one side of a cell to another side of a cell. Right. Something weird, I always find this kind of weird. Osmosis, when you've got, and this is what you should have seen with your potato. By the way, it should have been like this for the potato. So in a potato, here's a little potato slice, and here's some water that the potato's in. Oh, that should have been a container like this. Okay, here's the water inside the container. Because there is more salt and solute inside of this potato, there's also sugars, there's also other things that are dissolved in a potato. And there's not so much of that stuff on the outside. And this membrane right here in a potato is it's permeable to water, they say. It's permeable to water, so that means the water can go on either side. What do you think would have happened to this potato if there's hardly any solute on the outside? There might be a little bit. And there is tons of solute on the inside. You should have seen that this potato over time would have gotten water pumped in, well, not pumped in, but the water naturally goes to its lower concentration, which is inside this cell. So inside, or lots of cells in here, inside this potato, which has lots of cells and lots of solute and lots of things dissolved in it. This potato should have gotten over time bigger because water got put into it. The reason water went into it is because of osmosis. Okay, it went in because this water was going down its concentration gradient. It was very packed over here, outside here, and now it's going to a lower concentration of water relative to where it was. So this potato should have gotten bigger. You should have seen the mass got bigger. You should have seen the size got bigger if you would have measured it. Some people didn't measure it. Make sure you measure everything, everyone, when you're doing labs. Okay, so this is an example of osmosis. Another thing we can talk about here is that relative to this water outside, this potato is something we call hypertonic. Okay? Right. Think about what would happen if you have, like, on another kind of example here, the reverse example. Let's imagine now that you've got something else plopped into water. Let's say you've got, let's use the same potato. It's a smaller potato. It's got all of its solute out here. But now, on the outside you've got tons of salt dissolved, like more salt than this potato has inside of it, or more solute than this potato has inside of it. You really got to pack this thing full of solute to get more of the solute out here. For your experiments, it's kind of hard to get so many salt or sugar or whatever dissolved so that it's more concentrated than inside the potato. But I'm going to try to draw that right here. So I'm going to say there's more out here. Okay, more out here. Right. So there's more than in the inside. So what's going to happen to this water? This water is going to say, all right, well, it's pretty packed in here with water. I think there's less water out here. Oh, yeah, let's go outside. Okay, so the water goes out. The water goes down its concentration gradient. It's just diffusing. That's all it's doing. It's not thinking. It's just going down. It's like spreading out. Just like here, it's like spreading out. So over here, there's equal concentration of water. Just like over here, the water goes out of the cell. So if the water goes out of the cell, in this case, this thing is called hypotonic compared to the solution. And the result of this is going to be that it gets smaller, so it shrinks in this scenario. So it shrinks here, and over here it expands. All right. Wunderbar. The last thing I want to talk about is this idea. Well, not an idea. It's just kind of like a list of organelles and so forth. Okay, so if you don't know the functions, and I was a little concerned by some of your responses to the cell simile project, if you don't know the functions of cells, organelles, you need to review this. Oh, stop. Okay. You need to review this. Here we go. Organelle review page. All of these are great resources. Well, types of cells, you need to review these. If you don't know this, please read these. It's essential reading for this test. This is a great link. Okay, so sometimes things don't work all that great, but you can still find a way around it. Like in this case, it says you're trying to launch blah, 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 blah. Doesn't work. Well, I'm just going to click on it. Okay. Oh, there we go. Great. Now I've got this cell in front of me.
Lysosomes and other specialized vesicles.
Blah, blah, blah. So now I'm going to go to make the cell transparent, so now you can see what I'm pointing at. And if I go to one of these, oh, this is great.
The cell's nucleus stores and protects the organism's unique DNA.
This is fantastic. I don't know if you can hear this, but there's a narrator here telling me what this nucleus does. So it's pretty cool. Go through this. It goes through all the different organelles, the main organelles that we're going to be tested over. Go back to the modules. And I should have you take a look at this. Okay? This tells you what these organelles look like, so you're going to need to know what they look like and what they do, and in which cell they reside. So here, look at all the different organelles that an animal cell has. Look at the plant cell, all the organelles that it has. A good rule of thumb is that the plant has everything. Okay, so it doesn't really, whatever organelle I put out there, the plant is going to have it all, because plants have so much stuff. I wish we had so many things in our cells so that we could, make our own energy from sunlight and water. That would be awesome. We wouldn't have to eat. But now we have to eat plants, and so we can get our energy. The prokaryote or the bacterial cell has always the least amount of organelles. And here it shows all the organelles that a bacterial cell or a prokaryote has. Or I'm sorry, all the organelles that a prokaryote or bacterial cell has. It's the basic kind of cell. It's very primitive. Here it doesn't even have a nucleus around its DNA. It does have ribosomes. It has this thing called a plasmid right here, which is just a little kind of ring of DNA. We'll talk about that much more in detail later. They have these little formations called pili, which are extensions of this green thing called the plasma membrane. They also have this flagellum. Animal cells have this too. Some animal cells have this, so it's not just bacterial cells, but that's kind of good to know that bacterial cells have this. So bacterial cells have plasma membrane, cell wall, and this thing called a capsule as well, which protects its cell wall. Antibacterial soap and so forth, or just plain soap, breaks down this cell wall in this capsule, so washing your hands with soap does that pretty well. It has a cytoplasm as well. So if you commit these organelles to memory, and I'm afraid you're going to have to, that is all that bacterial cells have. And then the differences between the plants and the animal cells, you can see just by scrolling up and down with this right here, you can see what's different between them. I also gave you the answer key to a workbook problem, and I'll pull it up right now. That gives you a bit of a chart to refer to. I think that's it. Yeah. There it is. Okay, so on the very last page, I give you a bit of a chart. You can pause this right now and see that all of these organelles are in the plant cell. A few of them are in the prokaryote. You see that? Prokaryote has cell wall there. And then those other kinds of things. And the animal has most of this except for the cell wall and a chloroplast. Okay? So those are some of the tips I go through when I think about plants and animals. The main difference between plants and animals is their cell wall and their chloroplasts. Plants have everything, and prokaryotes, I remember that they have these things right here. Okay, that's all I have for you. Good luck studying. I'll be here if you have any questions. Bye.